Hello, everyone, and welcome to today's session. Thank you all very much for coming. It's my pleasure to welcome you to our webinar series on the future of treatment this month, part of our Living Well with Prostate Cancer webinar series. Today, we will be talking about demystifying prostate cancer clinical trials. And my name is David James, and I'm the Head of Patient Projects at Prostate Cancer Research. So first of all, I'd like to tell you a little bit about who we are. Prostate Cancer Research are a research-focused charity that's dedicated to improving and saving lives by developing new treatments for advanced prostate cancer. By funding novel and innovative projects at world-leading institutions across the UK, we're working towards breakthroughs in the prevention, diagnosis, and treatment of advanced prostate cancer, and ensuring better quality of life outcomes for patients. We're very proud to fund 15 research projects in Aberdeen, Cambridge, Cardiff, Glasgow, London, Newcastle, Norwich, and Oxford, focused on key areas such as bone metastases, radiotherapy, immunotherapy, and AI, with a shared aim of reducing morbidity, increasing survival rates, and improving quality of life outcomes for people with prostate cancer. Now, I'd like to take a few minutes to tell you a little bit about one of our research projects in this area. So launched in 2005, Stampede is one of the largest ongoing UK clinical trials into treating advanced prostate cancer. The trial aims to evaluate new treatment options and approaches and compare them to existing options. So far, more than 10,000 people have joined Stampede, and results from the trial have already changed the way that prostate cancer is treated. So PCR are funding a project led by Professor Gert Attard and Dr. Emily Grist, which is a subset of the Stampede trial. And this is looking into developing a test to help choose the best treatment for individual patients. Now, the problem lies in the fact that prostate cancer is the name used for all cancers that start in the prostate. However, prostate cancer tumors can be very different from each other. And this can make it difficult to know the best way to treat an individual's prostate cancer. Different cancers have unique features which may make them particularly suitable for a certain type of treatment. And then patients can be given treatments not suited to their individual cancer, or they may even be overtreated. So it may be possible to find patterns in the DNA of tumors that can be used to predict which treatment will work best for the individual patient. So Gert and Emily will analyze prostate tumor DNA taken from samples collected from men on the Stampede trial. They will identify patterns in the DNA and explore whether differences in tumor DNA can be matched with the patient's response to treatment. If successful, biopsies taken from patients during diagnosis could be tested for these patterns in order to determine the best treatment pathway for the individual. Whilst this project is still in the early stages, researchers are hoping that combining the results from this study with other Stampede projects could lead to real changes in the way prostate cancer is treated. So it's my pleasure to welcome you today to our webinar series, Living Well with Prostate Cancer, which we launched in collaboration with our good friends over at Tackle Prostate Cancer, who are the UK's National Federation of Prostate Cancer Patient Support Groups. Today, we'll be covering the important topic of clinical trials in our webinar called Demystifying Prostate Cancer Clinical Trials. So a few quick housekeeping points before we start this session. So for any of you who have joined us previously, you'll know, but all attendees, microphones are muted and cameras are off. And this is so that we can record the session and then share it with anyone who wasn't able to attend the session today, or for those of you who are interested in watching it back at some point in the future. There isn't a chat box function, but there is a Q&A box. So any questions you have during the session, feel free to type them in there. And then when we open up for questions at the end of the session, we'll do our best to get through them as best we can. So I'm very pleased to introduce you to our three speakers today. We have Professor Andrew Hughes, who is the Professor of Experimental Cancer Medicine team at the University of Manchester, and previously the clinical head of the Manchester Experimental Cancer Medicine Centre, which is one of the largest phase 1 units in the UK. And he is joined by Dr Nigel Brookes, who's the Director of Translational Science, and Stephen Salomon, who's the Clinical Trials Manager at biotechnology company CellCentric, which has a new drug in development called CCS 1477, which is for the treatment of patients with late-stage prostate cancer. So without further ado, it's my great pleasure to hand you over to Professor Hughes.
Thank you very much, David. Let's move now on to the first of our slides. And I'm delighted to be here today to address you. And David firstly asked if I would say a little word or two about myself and how the whole of my life has led up to this point today. So I was born in the first city of Liverpool, supporting the best football team in the world before getting an open scholarship to read medicine at Cambridge, where I intercalated that with a neuroscience PhD, looking, would you believe, into the drivers of male sexual behavior. We can have a Q&A of that at the end if you want to. But then I became a cardiologist in the NHS for a number of years before joining ICI Pharmaceuticals, as it was in the 1990s. And in my industry life, taught a lot about advanced cardiac life support to people within the industry, because I worked on a clinical pharmacology unit where we administered a whole host of drugs to particularly healthy volunteers to see whether they were worthwhile going into later phase testing. And I had the privilege of working on a number of drugs, including Seroquel for schizophrenia, Zomig for migraine, and Arimidex for breast cancer, before, in fact, 25 years ago, joining the oncology group at AstraZeneca and working on a number of products over those life cycles until I left AstraZeneca in 2015 when they moved to Cambridge as the global head of early clinical development. And I joined Manchester as a professor of experimental cancer medicine, where my research interests included continuing looking at phase 1 clinical trial agents, and we'll define that term in a few moments, and also helping quite a number of groups discharge their monies in terms of prioritizing research in clinical trials. And I'm often asked, why did I join cancer? And it's really 3 core reasons. Firstly, as we'll see in this mini lecture, it's a really, really challenging career. It's intellectually challenging, frustrating, but incredibly rewarding. But secondly, because we live in the Northwest and I work at the Christie and the University of Manchester, I'm really driven to try and make a difference to healthcare in this part of the world. And then clearly, all of us, I suspect, know people who we've lost prematurely to cancer. And that, again, underpins part of my motivation for working in this area. So what I'm going to cover in the next 30 minutes are 4 formidable challenges. Firstly, we'll talk a little bit about cancer to get into the zone, then focus on the key topic of this seminar, which is clinical trials. Now, look about the formidable challenge of personalizing medicine. We speak and hear a lot about that in the news, but actually it's incredibly difficult to operationalize in practice. And then I'll close with saying a few words about the very practical issue of the industry academic divide and how organizations like Prostate Cancer Research can actually close the gap in helping industry and academia work together. Let's start off with cancer. And a picture says 1,000 words, and the picture on the bottom left is a picture of cancer. It's a biopsy of cancer, and every black dot is a cancer cell that happens to be dividing, and it takes about 24 hours for a cancer cell to divide. So in this screenshot, about 30% of the cells are brown, therefore 30% of the cells are dividing, the other 70% are blue. They're just resting, waiting for their moment in the sun when they're going to divide. And that's really quite a high proportion of a tissue that's dividing. Most of our normal tissues are only turning over at 1 to 2%. Our gut, which is the most rapidly turning over tissue, about a quarter of our gut gets replaced every day, only has a proliferation rate of about 10, 15%. So for a cancer to be proliferating at 30% explains really why a lot of cancer patients get fatigued, because it's literally like being pregnant, but growing something within you that is foreign rather than made of you. And it's foreign because of the cartoon on the right-hand slide. This is a really, really simplification of cancer, and it underpins the formidable challenge to understand cancer, because there are a gazillion processes that go on to drive that increased proliferation that underpins cancer. So let's start with a poll, and I'm going to turn over to David to ask you 6 basic questions on cancer, so we can level set the audience of about 60 that have joined us on today's seminar. David, over to you for the 6 questions that we're going to ask. We're going to have about 1.5 minutes to answer these 6 questions. Don't analyze them too long, just put your immediate gut down and go for it. So the first question is, what % of people are going to get cancer during their lifetime? 1 in 10, 1 in 5, or 1 in 2? The next question is, how often does our body typically encounter a cancer cell? Is it once a day, once a month, once a week, or once a year? Again, moving on to question 3, how many different types of cancer do you think there are? We use one word to describe cancer, but in fact, there are a myriad of different cancer types. How many do you think there are? And then question number 4 is how many cancer treatments are under development right now? Do you think there are 100 new drugs undergoing investigation, 300, 600, or 900? And then the finances of developing a drug, how much do you think it costs? And mark your choice up there. And then when you start clinical trials for the very first human that ever receives the drug, what do you think the chances of that cancer medicine ever reaching you and I are as patients? 10%, 25%, 50 or 75%? So just spend another 60 seconds or so deciding on your own choice for that poll before I ask David to close it. Okay, 15 seconds, and then I'll ask David to close it in 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Thanks very much, David. If you can close it, and by the wonder of technology, I actually get the answers that you've all expressed on these questions. So 2/3 of you said that 1 in 2 of us were going to get cancer during our lifetime, and that indeed is the correct answer. On average, it's estimated that once a day, 1 cell in our body becomes cancerous, and therefore our immune systems do a great job in picking up a cancer cell that's become foreign to us and killing it. And 4 out of 5 of you were correct in declaring that response. Now, if you went on to the National Cancer Institute website, which is a group in the US that administers a lot of cancer care, they will say that there are 200 different types of cancer or near enough. And in fact, only 1/3 of you answered that correctly. Most of you, half of you thought that there were 400 different types of cancer, but there are in fact only about 200 or so classified as cancer. Now, there are right now 900 drugs undergoing development. So compared to our colleagues working in respiratory on diseases like asthma or chronic obstructive pulmonary disease, where there are only about 200 new drugs under investigation, we have the luxury in cancer of having almost 5 times that amount of new drugs, which is great news for cancer. But only 1/3 of you answered that correctly. And it costs about £1.5 billion to develop a drug, right away from the earliest phases of discovery, which we'll talk about in a moment, through clinical testing and to launch. It's roughly the same that NASA spends when it used to launch its space shuttle into space and back again. So it's £1.5 billion to develop a cancer drug, which is why this is not for the faint-hearted. And the industry as a whole develops 97% of cancer drugs because they have the deep pockets to do it. Only 1 in 4 of you got that correct as an answer. And then the chances of a cancer drug getting to market when the very first human is tested is only 10%. 1 in 10 chance of getting it to market. So there's a huge attrition rate in development, which is part of the reason why a cancer drug that makes it costs so much, because you've got to pay for the development of 9 drugs that don't make it. And that's something which is not widely appreciated, because you look at the eye-watering price of 1.5 billion and you think, my, how can you spend that much on 1 medicine? Actually, you're not. You're spending it on 10 medicines for 1 to make it. And that's where the funding for R&D, research and development, comes from. 2/3 of you actually entered that correctly. So, you know, great job on that. So that's our first poll of 6 questions on cancer, just to get us into the zone. Now, when we talk about cancer drugs broadly attack cancers in different ways. Firstly, and cytotoxics, the good old classic cytotoxics, used to do this. They just stop that cell division in its track, that 24-hour process that I described earlier, in different ways, either by damaging the DNA or not providing the DNA the material to replicate, or by stopping the cell dividing. Cytotoxic drugs stop cell division. More recently, we've actually got some drugs that help to kill a cancer cell. They tend to target themselves specifically to some of the weaknesses that cancer cells have and promote cell death. So it kills the cancer without killing the normal cells and thus you. The third way, and again, we've had a lot of this in the last decade or so, is if you can see those little black cancer cells, they all have furry coats on. And those furry coats help those cancer cells be protected from the immune system. They essentially cloak themselves in a Harry Potter invisible cloak. And the new drugs that are on the market that are immune checkpoint inhibitors or immune stimulants, they go by different names, tend to decloak the cancer. And so suddenly the immune system wakes up and says, aha, and now I can see you, now I can spot you, and now I'll destroy you. And that's part of the great revolution in immune inhibitors that have come onto the market in the last 10 years. We haven't really got a drug yet that stops cancer cells getting into the bloodstream and spreading in the bloodstream, but that's certainly been a fertile source of research over the last 20 or so years. But we do have drugs that now can selectively destroy the blood vessels to a cancer. Would you believe it, that the blood vessels to a cancer are subtly different than the blood vessels that feed our retinas or our brains or our kidneys? And very cleverly, drug developers have managed to find a way that they can selectively stop the blood supply to a cancer, and so it therefore destroys itself because of lack of nutrients and oxygen supply, without actually doing damages to our retinas and to our brains and to our kidneys. Let's talk now about clinical trials. And it's a formidable challenge because as we look at 30,000 feet into the stages of drug development, quite a number of years are spent before clinical trials even start. They're spent in preclinical or sometimes non-clinical testing, as it's called. That's all done in animal models or indeed cell lines or in cells that have been derived from cancer patients. And so these in vitro, which are done in a test tube, or in vivo, which are done in animal models themselves, these tests demonstrate that the drug has good anti-cancer activity. And then, as we'll see in a few moments, some toxicology tests are done in order that we can safely then start the clinical testing. The first phase of clinical testing, not surprisingly, is called phase 1. And that's because the first thing we need to characterize is the safety and checking for safety in that phase 1 study. And that will be the focus of the majority of this discussion because time doesn't permit us to talk about the other phases of clinical trials, but it tends to take a year to 2 years worth of testing. Phase 2, or the second phase of clinical testing, is checking for activity, for clinical efficacy, that the drug shrinks tumors or prolongs survival or delays the time that it takes for cancers to progress and become metastatic and spread around the body. And again, because that takes time as a process, then these clinical trials themselves take 2 to 3 years typically to do. In phase 3, or the third phase of clinical testing, this is when you need to generate the data to demonstrate to payers and to regulatory authorities that your drug is better than the other options that are out there. Unlike some other areas where equivalent drugs are often licensed, in cancer, the regulatory authorities and payers will only tend to rightly, in my view, pay and approve drugs that are better than things that are already existing. This is such a desperate disease that why would you want to pay for something that is just as good as something else? And so phase 3 is about demonstrating that your approach is superior to the approach that you're trying to beat on the market. And then after launch, which is done in phase 3, phase 4 tends to be the long-term surveillance for rare and unknown safety effects. And thalidomide is a great drug, which was launched for sleeping and insomnia in pregnancy. And it was only through this phase 4 surveillance system that the thalidomide babies were picked up as a result of that. It wasn't a side effect that was picked up during clinical testing. And that's why the number of successful drugs diminish and have those high attrition rates that we spoke earlier. But as I say, the purpose of today's seminar is really to focus just on phase 1, because actually the UK is pretty good at phase 1 cancer trials in particular. And that's because over a decade ago, the government, along with colleagues in Cancer Research UK in their wisdom, started to invest in a number of key cancer centres around the UK. They're called out on this slide. And over the last decade, a significant amount of funding has been given to these cancer centers to employ nurses and doctors and clinical trial staff to make sure that we can punch above our weight as a nation in doing phase 1 clinical trials. And so, in fact, we're only second now to the US in doing phase 1 cancer trials. And the reason why these centers were selected is that the heat map on the right just shows where the patients to these centers are now recruited from. And actually about 90% of the population is 50 or so miles from one of these conurbations. So as we'll come to, there's always a good clinical trial that's potentially close to you. And so that leads us on to where to go to find information. And by far and away, the best information on clinical trials is held on this US National Library of Medicine called clinicaltrials.gov. And the reason why it's the best website for finding a clinical trial, or the definitive website for finding a clinical trial, is because every sponsor of a clinical trial by law now has to put their trial onto this website so that no two sponsors do an identical study. And so we're efficient in our use of patients rather than asking patients to recruit for a study that's going on somewhere else in the world. And so every sponsor of a clinical trial loads their clinical trial onto this website. If you were to go onto the search engine on this website and just type in, as I did, prostate cancer metastatic, so to look for trials in patients where the prostate cancer had spread beyond the gland into, for example, the bone or liver or lungs, and I wanted to find a site within 50 miles of London that was doing a prostate cancer study, as you can see by the search, up pops a number of clinical trials. I've chosen number 5, which is a trial of pembrolizumab, which is an immune inhibitor that I described earlier, looking at whether it works in men with metastatic castrate-resistant prostate cancer. I see that it's being done at the Royal Marsden, and then I can scroll down and look at what's called the inclusion criteria or the eligibility criteria. Most trials actually have about 60 or so inclusion and exclusion criteria. Now, some of those you need some medical opinion in order to understand, but others, male, aged 18 and over, any of us can figure those things out. So at least you can get a first pass as to whether you might, or a friend of yours, might be eligible for these trials. So once you've looked at all of your data, then you can essentially ask your referring physician to refer you to the center, whether that be your GP or oncologist, or your friend's GP and oncologist. But as mentioned, this is not, whilst it's a definitive website, it's not a user-friendly website. And David assures me that prostate cancer research will hopefully be putting forward a website that is more cancer patient-friendly. So the main purpose of a phase 1 study is to look for and to characterize the key safety findings that are likely to cause an issue to your drug, to kill it or to restrict its label, or to have to result in monitoring its drug levels. And typically different cohorts of patients are receiving increasing doses until, in this example, cohort 5 is the highest dose that is well tolerated by the patients. And so we have a little look-see at an expansion cohort to see and confirm that it can indeed be well tolerated. And the goal of that phase 1 study is, as I say, to characterize the toxicity, the human safety of the drug, the pharmacodynamics, what the drug does to the body, and also the pharmacokinetics, what the body does to the drug. And we'll spend now a few moments on each of these 3 aspects of tolerability, pharmacodynamics, and pharmacokinetics to get a better understanding as to phase 1. Let's start on toxicity. And the key goal here is to understand how good are the animal species in predicting human toxicity. So David, if you can launch the poll, there are 5 questions that we'll have to see how good are the animal models. We tend to do drug testing in a rodent species like a mouse or a rat, and a non-rodent species like dog or a monkey. And so the first question is, what % of human toxicities that were seen in the animal species do you actually translate to man? Are the animal species misleading us at 30%, or are they really predictive up at 90%? So how good are animal species at picking up human toxicity? The second question is, if you had to just choose one species, the dog, monkey, rat, or mouse, which would it be to predict most human toxicities? Thirdly, some of our body systems are more rather than less predictive. Do you think the skin of an animal is more like the skin of a human. The blood of an animal is more like the blood of a human. The gut of an animal is more like a gut of a human, or the brain and nervous system of an animal is more like a human brain and nervous system. Question number 4 is, when do you think these bad toxicities that kill a drug, limit the label, or cause drug monitoring are actually revealed? Is it phase 1, phase 2, phase 3, or phase 4 that we defined just a few slides back? And then lastly, most drugs are tested for a month in these non-rodent and rodent species. What percent of human toxicities that are going to kill your drug are actually picked up by the animal species? 95%, 85%, 75%, or 65%? Let's just have another 15 or so seconds on those 5 questions. I'm afraid there are no prizes. It just gets you into the zone of the next few slides. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. And if you can stop the poll, David, thank you. Let's have a look now at the answers. Okay, so the first question, what percent of human toxicities were seen in the animal species prior to human testing? In fact, 70% of human toxicities are picked up in animal testing. The non-rodent on their own picks up 63, the rodent picks up about 43, both species together pick up 71%, and no species only pick up, so 29% of all toxicities you don't actually see in the preclinical models. And only a third of you got that question right. Now, I've got to be careful on this one because men are always referred to as one of these animals. And in fact, the dog is the most predictive. If you just had to choose one species, and a lot of you, actually 50% of you put monkey down, 8% put rat, only 2% of you called out the dog. And in fact, the dog is actually the most predictive of all animal species for predicting human species. And in fact, animals' skin are very different from human skin. That's not surprising, but the blood systems are actually very, very similar. So if something was seen causing, for example, anemia or reductions in white cells or increases in platelets in the blood system of an animal, then it's most likely actually to predict to human species. And 46% of you got that one correct. 4% of you, interestingly, thought that animal skin and fur was very much like human skin. Perhaps we better send you on a training seminar series for that on the next. But let's move on. 61% of all major human toxicities come to light during phase 1. And again, a third of you got that answer right, which is by the time you leave phase 1 clinical trials, only 1 in 3 drug developments are you ever going to encounter something which is going to kill your drug due to toxicity after phase 1 clinical testing, provided phase 1 clinical testing has been done well. And then to question number 5, when we do a standard 1-month test, 95% of all toxicities that are going to be picked up by the animal species have been picked up. And 20% of you, 1 in 5 of you, got this right. And this is an important slide because before this data was done in 1996, the Japanese were doing 6-month studies and the Americans and British were doing 1-month studies, and the Japanese were saying, you know, you're cutting corners and you should be exposing animals, more animals for longer. And the Americans and British were saying, actually, we think we've got it right. And in fact, this was the slide that switched the Japanese and saved huge amounts of animals in toxicity testing. So let's now move on to talk about pharmacodynamics. This is what we defined as what does the drug do to the body? And we can pick this up really in one of 3 ways. We can either take a piece of tissue from the tumor and look at it down a microscope to see if the drug has done anything to the microscope of the tumor, or we can take a blood sample and we can see whether anything that the tumor releases into the blood, like circulating tumor DNA or it's circulating tumor cells, or blood markers like prostate specific antigen, PSA, we can measure those things as a biomarker of wealth and health of the tumor itself. Or we can directly image it, CT scans, MRI scans, PET scans. Again, we can see whether the tumor is living and breathing well, what its blood supply is like and how big it is. So those are the three ways we tend to see whether the drug does anything to the tumor in a phase 1 clinical test. And the benefit of that is in a real example when I worked at AstraZeneca, where 1 in 10 patients in green had a good response. 2 out of 10 patients, it was like giving distilled water, and 7 out of 10 for 3 months or so that they were on the trial just had stable disease. And everybody said, oh, well, is that just because the 7 would have had stable disease anyway for those 3 months, and therefore the drug is doing nothing? Or actually, because 1 person did respond, maybe the drug is doing something. And so to help with this debate, we took a pre-dose set of biopsies to look at the drug target effect in brown, or the cell division I described earlier in the bottom panel. And then we biopsied when they were on drug, and you can see that the brown has gone and turned to blue, and those dots have gone. And again, they've stopped cell division. So we knew absolutely that the drug was getting to the tumor in those patients with stable disease. And this wasn't just serendipity, but it was a real drug effect, that these patients got drug into their circulation. The drug got into the tumor, it changed the tumor and produced a stable disease. Let's close out this section now by looking at pharmacokinetics, which I defined earlier as what the body does to the drug. And when we inject a drug intravenously, the concentration of the drug falls over time. If it's got a long half-life, it falls slower over time. If it's got a short half-life, like aspirin with a 2-hour half-life, the concentration falls very rapidly. If you were to dose it orally, it takes a little while for the concentration to increase first. It peaks at a maximum concentration at a time of maximum concentration, and then again decreases over time. So understanding the pharmacokinetics of a drug helps to understand how frequently you need to give the drug. If it's got a very short half-life, you might need to give the drug 4 times a day, as some cancer medicines are given. If it's got a very long half-life, you might only need to give it once every 3 weeks, like some cancer medicines are given. So the pharmacokinetics and understanding how frequently it needs to be given is, again, a key thing that we need to figure out in phase 1. So let's move on to the third formidable challenge, which is personalizing medicine. And that's a formidable challenge because from a distance, all snowflakes look like snow. We use one word, snow, to describe millions and gazillions of different size and shaped snowflakes. And equally, from a distance, we say that all patients have cancer, and we use this unifying single term of cancer to actually describe millions of diseases, because in a very nice study that looked at 57 women who presented with breast cancer, and they looked at the DNA content of those 57 women, actually no 2 patients had the same genetic profile. These are just some illustrative pictures of each of the patients, and they are just very, very different snowflakes, the 57 women. And in fact, those 57 women had 12 million alterations identified in their DNA. So really, we should move away from just looking at cancer as a picture and saying, well, you have prostate cancer or breast cancer or lung cancer, and actually looking at the DNA of each cancer. And in the same way that we as people are unique, every person's cancer is unique. It has a different makeup of DNA mutations. And so the challenge now is going from a one-size-fits-all medicine to actually a situation where we do look at the DNA, we do molecularly profile patients, and then we match the right medicine to the right DNA profile of that patient. And the importance of this has transformed phase 1 testing, where previously only 1 in 20 patients that got onto a phase 1 trial would benefit. Now it's closer to 1 in 2. Patients like this one. This was a young patient with lung cancer, and you can see on the CT scan that the left lung, the lung on the left-hand side of the picture, which is in black, that's a good and healthy lung, is almost obliterated on the right-hand side of the picture in white, because the cancer is filling up the vast majority of the lung. Just after 1 month of being treated with an agent that was targeted to a genetic change called NTRK in their gene profile, this was the CT scan where you can start to see the patient's lung on the opposite side turned black, which is a normal aerated lung full of air and oxygen. And in fact, even a year later, the picture was improving, where the lung was essentially now back to normal, where there had been an 80% clearance of tumor, 50% clearance after just a month of dosing, which is remarkable by matching the right drug to the right genetic makeup of the patient. So going forwards, getting our genetic profile for our cancers is really quite key. And the NHS has made significant strides in the last year by setting up 6 genetic testing hubs across the UK, where hopefully over the course of the next 5 years, anybody with cancer will now get their genetic code. Let's finish off this section before I hand over to the CellCentric team to talk about the specific trial that you've worked with us to generate in talking about this challenge of working between industry and academia. You heard me say earlier that 90%, 97% in fact, of all cancer treatments originate from industry because they need the deep pockets of £1.5 billion in order to develop a cancer drug. Whereas academia and the NHS have all the patients that are required to do the studies. And so there is, whether we like it or not, a difference in territory, a difference in ground, a bridge, if you want, it needs to be established between these two communities where there are misconceptions on both sides of the house. Having worked in industry, there's a misperception that it's just a pot of gold. And having worked there for 25 years, there was no year that I worked where I had enough money to do everything I wanted. We always had to prioritize. Equally, there's a perception that we're preventing, or industry's preventing, publication of data. And that certainly was true 20 years ago. But now there's transparency and in fact legal constraints that within a year of the clinical trial being completed, you need to publish the results. That's no longer true, and industry can't hide them. The strength of industry is in its decision making. It comes to decisions and then ruthlessly implements those decisions. In academia, sometimes I found it very difficult to find the decision maker because the decision maker doesn't realize that they actually have the accountability for making the decision. That's really quite transparent in industry and one of the strengths of industry. But one of the constrainers of industry is that very much it does things to its priorities, and those priorities can change. So GlaxoSmithKline, some years ago, didn't think that cancer and developing cancer drugs was a priority. Now it does. And so it lost a whole load of capability and expertise by a priority decision 7 to 10 years ago that it now has to reverse. And that's really frustrating for those that are in that sector. Equally, it has to make a return on investment from what it invests. Otherwise, clearly, no new medicines will be developed. And that's an issue for some of the rarer cancers where it's much more difficult to get a return on that investment. So the rarer cancers are often done and boosted by funding from the charity sector. In industry, in academia, some have a misperception of academia that it's jobs for life. And that's certainly not true, or indeed that nothing is ever measured in academia. There's no metrics. And again, having had annual performance reviews at the University of Manchester, I can tell you that we are absolutely measured by our outputs. One of the strengths of academia is you do have greater degrees of freedom to work on these more curious and smaller prevalence diseases that industry might not be interested in because of lack of return of investment. But you do have constraints. Sometimes if you think of a good idea, it's you left to execute that good idea. You don't have resource around to help you. And equally, because of that, and because of sometimes the secrecy that exists, often the left and right hand don't know what each are doing within that ecosystem of academia. So there are differences between these two communities that place people like prostate cancer research can do a key role in bridging. And when I was in AstraZeneca, we recognized that a great collaboration is not just about great science, but it's also about actively engaging and managing that relationship across the divide. And so we put together a 5-point charter: respond quickly, don't ignore emails from the other side, respond within 24 hours, have one point of contact so that people aren't just being bounced from one call center to another call center. When things go wrong, look upon that as an opportunity to build trust and to work through things that go wrong rather than just blaming the other side. make sure that protocols do fit the world in which they're trying to execute. And again, this is a key role that you'll hear later prostate cancer research can play in the CellCentric protocol development. And then lastly, provide support throughout the relationship. Just don't have a great honeymoon and then a divorce. Actually work through the challenges as and when they arrive. So in conclusion, these are the 4 take-home messages from me. If you're wanting to look for a clinical trial for yourself and others, best place to go for is clinicaltrials.gov. I accept that the search engines aren't that patient friendly, but actually it's the definitive location to find a trial that then you can get your doctor to refer you for if you're interested in that. I've told you that in phase 1, which is the first phase of clinical testing, the 3 key goals are to characterize the safety profile, understand what the body does to the drug, and understand also what the drug does to the body, so-called pharmacodynamics. I've also taught you that instead of getting diagnosed with breast cancer or prostate cancer or pancreatic cancer, actually it's better to get diagnosed with what your genetic profile is in your cancer, because that can guide better personalized treatments, which have a better response rate than giving it to everyone in a carpet bombing approach. And then lastly, don't underestimate the very human challenges of this industry, academic, medical interactions. And that's where the glue that PCR can bring is key. So with that, I'll hand over to Nigel, who will tell you a little bit more about specifically the CellCentric trial, which is being executed in centers like the Christie and the others that I showed you on a previous slide across the Experimental Cancer Medicine Center network, and to which prostate cancer research has really helped shape the protocol to make it real for the real world. Nigel.
Thank you. I'll just wait while David changes the slides. There we are. Okay. So my name is Nigel Brookes. I'm the Development Director at CellCentric, and together with my colleague Stephen, who's a Clinical Trials Manager, we're going to give you an introduction to CellCentric, tell you a little bit about who we are and what we do, tell you a little bit about how we discover and develop a new medicine, and importantly, how we get that into a clinical trial. And then Stephen's going to discuss a terrific partnership that we've had with prostate cancer research to gain a much better understanding of patients' experiences really in clinical trials and on various treatments. That's really allowing us to do things differently, and certainly that's been a fantastic collaboration. So on the next slide,
Nigel, can I just jump in? Can you see the full screen or the presenter version?
I see the full screen.
Fantastic. Okay, great. Thank you.
Great. So on the next slide, just a little bit about myself. I would regard myself as a biologist, and I did a biology degree and PhD at University of Nottingham. And then I, after a short period of time in Canada doing a postdoctoral fellowship, moved to Edinburgh at one of the Medical Research Council research institutes, where I spent 10 years in academic research, understanding the causes and prevention of preterm birth. Quite different really from where I've ended up, but based on a sound understanding of the biology of various diseases. So moving from the MRC, I joined AstraZeneca and spent 20 years in a variety of different roles in drug discovery and development, all in oncology research. And I was very fortunate to head a cross-functional AZ prostate disease area team, which really set the strategy for what AstraZeneca should be doing to maximize patient benefit in prostate cancer through new medicines. And after 20 years with AstraZeneca, I had a great opportunity to move from a huge organization like AZ to a small UK-based biotech, CellCentric, and have been working with them ever since on the development of CCS1477, our drug for patients with prostate cancer. So on the next slide, just a little bit around CellCentric itself, an introduction to CellCentric. As I say, we're a UK-based biotech company. We're very science-driven, but very patient-centric. And in terms of the science, that really came out of academic research at the University of Cambridge, and the company was founded by Will West and Azim Sorani, who was a pioneer in a process called epigenetics, which is really how the structure of DNA modulates the activation of various genes. And through a number of different targets that were evaluated, we have focused wholly on one particular target called P300-CBP and developed this new drug, this new medicine, CCS1477. And we're very focused on the unmet needs, patient unmet needs. And we're exploring this drug's opportunity in 3 different areas. I guess most relevant to the discussion today is in prostate cancer, but also in certain blood cancers and certain other tumours that have genetic alterations that may make those tumours more responsive to the drug. So on the next slide, I'm just going to take you through very quickly about how we go about developing a new medicine in an organisation such as CellCentric. And it's very focused really on understanding unmet need. And that, in my mind, comes from 3 different sources. Firstly, it's very important to understand what the patient is telling us is the unmet need, where the need is. It's very important for us, secondly, to understand what the physicians sees as being the unmet need, what is missing from that physician's armory of medicines that is a gap that we can fill. And the third is really to understand the payers' need. What are they prepared to pay for a new medicine in terms of improving quality and quantity of life? And then underpinning all of that is actually a really deep understanding of the biology of disease. We can identify all this unmet need, but actually the biology isn't applicable to addressing this. And in respect to prostate cancer, all of those 4 things really align very nicely, particularly in late stage metastatic castrate-resistant prostate cancer, where, as you will all be, I'm sure, aware, patients who are treated with anti-hormonal agents such as abiraterone and enzalutamide, these are the so-called second-generation anti-androgen therapies. Regrettably, patients often relapse on those therapies, and that's because the tumours become resistant to those therapies. And regrettably, again, there is no real treatment for those patients, so there's an unmet need in patients that have progressed on those endocrine therapies. And we understand a lot about the biology of why those patients progress and why those tumours become resistant. And we know that it's because the androgen receptor is still firing away. The androgen receptor is the protein that mediates the effects of testosterone. We know that that is firing away to cause the tumours to continue to proliferate and progress. But the androgen receptor is no longer responding to these anti-androgen drugs. And so we need a new approach to how we might block the action of the androgen receptor. And so if you'd let's go to the next one, David. What we've done is to look at this new approach, and we've found 2 proteins called P300-CBP, which are analogous to the accelerator pedal in a car, and they are accelerating the activity of the androgen receptor in this late stage metastatic castrate-resistant prostate cancer. And if we can find a way of taking a foot off the accelerator, we can slow down the proliferation of these cells in this late stage setting. So we've developed an inhibitor of the accelerator pedal. It's a small molecule inhibitor that inhibits the activity of P300-CBP. It's very potent and very selective. And it's actually quite conventional in terms of how it's given. So it's given as a capsule orally. It's not dissimilar. The capsule is not dissimilar to a paracetamol capsule. And so we've developed this compound now, and we're in phase 1 clinical trials at the moment. And the next thing I'm going to describe to you is how we actually go about doing those clinical trials. So on the next slide, before I do that, I forgot to say we're going to have a quick poll, actually. So I'm going to hand over to David, who's going to just introduce the next poll. So it's quite a simple question, really, and it's really just to gauge how many of you have taken part in a clinical trial before. So there are 4 answers there. And if we just take 10, 15 seconds to answer that, and then we'll come back. Okay, I think given that it's quite a simple question, we probably should have got some answers to that now. Excellent. So the majority of you have not taken part in a clinical trial. That's about 80% of you. But 10% of you have either been in a trial in the past or are currently in one. So I think on that basis, we can say that, you know, it is an area that maybe is not as familiar to most people. And hopefully this seminar and this webinar rather is hopefully helping to demystify some of that. How do we begin clinical testing? So we've now been through the process of developing our new medicine, and we need to test that drug. There's a number of enabling steps that need to happen before we start a clinical trial. And the first one is actually to make more of the drug. So up to now, we've made about a tablespoon of the drug, and now we need to make a bag of sugars worth of the drug, you know, in kilogram quantities, to allow us to be able to then do appropriate toxicology testing, but also take it into the clinic. We need to find a way of formulating the drug. And as I said, we've formulated our drug as a capsule, but it could have been as a tablet, or it could have been as a mix and drink formulation in a phase 1 setting. Most importantly, we have to also understand the safety of the drug and whether it's going to be safe to administer to humans and at what dose would we administer it to humans. And so we do a number of toxicology testing studies as described by Andrew already. So those are the kind of enabling studies, and then we go through a process of clinical trial setup. And the first thing we have to do is to write a clinical protocol. And that's a pretty hefty document, about 150 pages worth, describing in great detail exactly how the clinical trial is designed and will be run. Accompanying that is another very hefty document called an investigator brochure, which is for the investigators, the clinicians who are going to administer the drug, to understand how the drug works, a lot about its pharmacokinetics, and a lot about its safety. We develop a patient information sheet, which is for you as patients to understand exactly what the trial is all about and what is involved with you joining a trial. And then we have to have a patient-informed consent, where you consent to be part of a clinical trial. And all of this paperwork, along with a lot of other pieces of paperwork, are sent for both regulatory and ethics approval, and that is conducted in parallel. And we go through a process of questions and answers with the regulatory and ethics authorities, and so we end up in a position, hopefully, where that is approved. And then the next step for us is really to identify our chief investigator. So we need a head honcho who's going to run the clinical trial for us. And we were extremely fortunate at CellCentric in having Professor Johan de Bono as our chief investigator. He's from the Institute of Cancer Research, Royal Marsden Hospital, and is a world-leading expert in prostate cancer clinical trials. And alongside Johan de Bono, we have Kevin Kelly, who is our chief investigator in the United States. So we have to then select site. We've got our chief investigator. We have to select sites, and so we have selected a number of sites in the UK, in Europe, and also in the US. We have to contract with those sites to agree all the financial relationships, and then we have to set those sites up and initiate them and train them into how to run our clinical trial. So before a single capsule has reached a patient, there's an awful lot of work that goes on to get us to that stage. And then on my final slide, really, we go into the kind of operational mode on the next slide. And the operational mode is now we are administering the drug to patients. We need to be very focused on how most efficiently to run that clinical trial. And so we have a group of people illustrated here. These are real people at CellCentric doing this clinical trial, led on the top left-hand side by Karen Clegg, who's our operations director, clinical operations director. And they're addressing all sorts of different aspects of the clinical trial, from the patient enrollment to maintaining all the data in a file, to the very important part of safety monitoring and ensuring that every patient is kept safe. And that's looked after by our Chief Medical Officer, Thomas Nurowski. We have a safety review committee that meets every 2 weeks to discuss the safety aspects of the clinical trial. A number of samples are collected, and Stephen's job is to move samples from one place to the other on multiple occasions to allow us to be able to measure things like the blood concentration of the drug in response to the administration of the drug. And then finally, we get a lot of data, safety, PK, and efficacy data that we have to analyze. And so this clinical operation team is responsible for all of those aspects. So it's been a very, very quick rush through, but I hope I've been able to describe to you how we take unmet need, develop new medicines, and get them into clinical trials, and then operationalize those clinical trials at an organization like CellCentric. And so I'm now going to hand over to Stephen for the final part of our webinar presentation.
Thanks, Nigel. So hi, everyone. My name is Stephen Salomon, and as Nigel said, I'm a clinical trials manager at CellCentric. Just a bit about me. I did my studies in Montreal at McGill University in molecular biology and neuroscience, followed with a master's research project. And then after that, I got more involved on the clinical side and worked at the McGill University Health Center, where our diagnostic lab was providing molecular testing service to the hospital for certain neuromuscular disorders. I moved more into interacting with patients more as a clinical trials coordinator in a hospital where I enrolled patients on clinical trials and followed them through their treatments. Currently, I'm now a manager at CellCentric, and where I work on the day-to-day clinical operations of our trial. This involves communicating with the hospitals that are recruiting patients on our study and making sure everything runs smoothly, from such things as samples being collected from the patients and coordinating the analysis of these samples. Next slide. So as a biotech company, we continuously ask ourselves, why do we do what we do? And usually the answer is always that we want to improve the outcome and quality of life for all our patients affected with prostate cancer, especially late-stage prostate cancer. And so the question is, how can we do this? How can we improve the quality of life for patients? And the best way is to hear directly from patients. And this can help in the initial early stages in the planning of drug development, to the design of the clinical trial, and even modifications or little changes that we can do during the trial while the patient is on it. Something to try to make it more patient-oriented. As a biotech company, we don't have necessarily the right means or tools in place to engage with patients. So how can we hear from patients? And we can do this through collaborations with groups such as the Prostate Cancer Research, who strive every day to make patients' voices heard. And this will help us to better understand a patient's experience on treatment as part of a trial or not, and possibly orient our trials and all trials more towards patient needs. Next. This need of patient engagement initiated really with a partnership between PCR and CellCentric. The partnership combines basically our knowledge in the biotech area and the clinical trial area, and PCR's close connection with patients, carers, and family members. In order to better understand the patient's experience in treatment or on a trial, we need to gather a lot of this information directly from you, the patient. And our partnership with PCR has led to various patient engagement activities where we can collect firsthand knowledge of patient experience. And this has happened through survey questionnaires, focus group discussions, and even speaking directly with patients. Next slide. And from these, the PCR actually was able to, they assembled a report on these patient engagement activities. In this kind of collaboration, we sought to understand what patients think about clinical trials and learn from their experiences on a trial, or learn from their experiences if they haven't been on a trial. So what our collaboration found from the report was that better information about clinical trials needs to be provided to patients. Patients want to be more supported throughout the trial, such as more contact with consultants or the clinical team, nurses, dietitians, and so forth. There could be a more focus on patients' quality of life when designing the trial, such as issues that relate to quality of life, such as traveling to hospital, the frequency of visits, providing quality contact, and then quality of life surveys, maybe while on the trial, to help to understand the patient's experience a bit more. I think making things simpler for patients as well, even regarding administration of medication, if there was a possibility of different choices. Next slide. Our collaboration and the findings in the report helped PCR create what's known as the info pool. And the info pool is a patient information and education platform. It has been developed by, with, and for patients to address all the unmet needs and challenges of people affected with prostate cancer. And now it's a continuous process where the info pool is continually being developed and could be used, will be used as more of an interactive forum for patients, where patients' voices can be heard, and is a place where patients can shape the latest research. They can voice their opinion, how some research projects that are currently going on. Next slide. I think some of the main key points that we've learned from our collaboration with the PCR is medication in the form of a pill, such as CCS 1477, is the least likely type of administration to stop patients from joining a clinical trial. Webinars are very helpful to patients and to the public. Hence, this is the one thing that we're doing today to help. We've learned that patients would like maybe some diary cards or apps that allow them to describe some of their good, bad experiences on a daily basis while on a trial. And also questionnaires, also known as patient-reported outcomes related to quality of life or pain, would be helpful and insightful for both the patient and the sponsor. And some of these questions, we've discussed internally, and we plan on adding this as part of our trial. Ultimately, we value your thoughts and experiences, and they are extremely important to us, so that we can understand how we can make your experience of a trial easier to improve outcome and quality of life. Next slide. I just wanted to kind of just summarize, and just in general, is that I think knowledge is key. We as a biotech company need to be a bit more patient-centric. Knowledge is key for all biotech companies to learn about patient experiences. And knowledge is key for patients to learn more about the disease itself and learn about clinical trials. And I just wanted to finish something that I always found interesting in that public awareness campaign for patients and general public have been a great way to grab people's attention, which kind of leads them to wanting to know more, being informed about it. Such examples included ALS, where they did the Ice Bucket Challenge, sarcoma, where they sarcoma is usually characterized as a lump on your body, and to make people more aware, they used an analogy of a golf ball, where their motto was "size does matter," meaning if it's anything larger than a golf ball, you better get it checked out. Breast cancer, the public awareness campaign they used was Know Your Lemons, and I think most of us know probably prostate cancer and Movember. And the No Shave November. Next slide is our last slide. Just a bit of our top tips that I could just briefly go through. I think number one is best place to look for clinical trials for yourself, for friends and family. It's clinicaltrials.gov, as Andrew mentioned, because you will often know more than your doctor when you're looking up some of these things. Number two, if you see a clinical trial that you think you may wish to join, then discuss this with your GP or oncology physician. And number three, the value of getting patient insight into the design of our clinical protocols cannot be understated. It grounds clinical trials in the real world, as we really value patient, your knowledge from your experience. Thank you.
Thank you very much, Steve, and thank you, Nigel and Andrew, for all of your presentations. They were very interesting and showed your different experiences from sort of your different points of view that hopefully have really helped people get an understanding of how clinical trials in the early stages work, and how these sort of very complex processes are put together. We had some questions coming in, and I think the first one is really actually from me was I get to cheat and jump straight to the head of the queue. Obviously, Andrew, you showed the sort of clinicaltrials.gov website, and if I recall, we sort of looked at the search bar, and you had sort of the inclusion criteria and the contact information. If someone wants to sort of speak to their GP or oncology team about a trial, so what do you recommend? Is maybe taking a sort of printout of those kind of key parts, or what should they bring to sort of generate this discussion? Because I know, of course, depending where you are, many GPs or oncologists may not be aware of the trial. So what would be most helpful for people to bring along to those sessions?
Yeah, definitely to print out three key things. Firstly, just the title of the trial, which you'll find on the website. Secondly, the inclusion and exclusion criteria, because your GP might be able to know, for example, that you're not eligible to that trial because one or other a blood test is going to be out of range, which you wouldn't know yourself by just looking at the website. And then the third key piece of information is the contact details for the site, because then the GP or your treating oncologist will be able to write a referral letter saying, they've looked at the trial, they've looked at the criteria, they think that you are eligible or your friend is eligible for the trial. So they've done an initial medical triage to make sure that in their view, you are a potential candidate. And then they would write a letter of referral, which in many ways, the receiving site is both grateful and duty bound to consider. So they would then call you up for either a telephone interview to ask you a little bit more about your interest in the trial, maybe clarify some other questions that relate to the eligibility criteria. But in essence, your GP or treating oncologist, they need that information to refer you to the site.
Fantastic. Thank you so much for that. There was a question, Andrew, as well for you. It was actually back on, you showed those lungs in the sort of personalized medicine section. It's a question actually around the lung on the left, which seemed to sort of be okay. Why did that also seem to improve during the process of the treatment?
So the lung on the left was the one that was full of cancer to begin with. The one on the right was actually pretty normal. There might have been a little bit of cancer around the outside, but it was the lung. No, hang on a sec. I'm just clarifying in the asker left and right, because rather confusingly, when a scan is done, what was on the image on the left-hand slide was actually the patient's right lung. It's just medical convention to confuse everybody, I'm afraid. So the lung on the left in the picture was the patient's right lung. Their right lung was predominantly normal. It was the left lung, the lung that their heart was on the side of, and therefore to the right of the picture, that was riddled with cancer. And that was the lung that basically had the 70–80% decrease in cancer over the year. It was down to a half of that so it may have simply confused you, I'm afraid, that medical images, they show the right of a person on the left and the left of the person on the right. So when I was describing left and right, I maybe have misled some of your listeners.
No, sorry, actually the question didn't ask that. I put in the left, so you confused me, I'm afraid. But I think the question was probably more, from my recollection, the lung, the black lung was the good lung, correct?
Yep.
And in the first image, it's it looks like a lung, but by the last image, sort of 6 months later, it's really filled out, right down. It's kind of completely black. So it would seem like that lung had also improved. And I think the question was why that lung had improved, unless perhaps there was some cancer that maybe had spread there, but it wasn't as much.
No, there was also some cancer in that good lung, and it tended to be around the edge of the lung. So when the image is done initially, it looks black, but not particularly clear black. It looks a kind of translucent black. And that's whereas you clear the cancer from the surrounding layer of the lung, the blackness becomes, crisper over time. So there was some effect on that other lung. Yeah.
Sure. All right.
Well spotted. Whoever spotted it.
There's a question, I think, for you, Nigel. This question is CCS 1477 the same as P300 CBP?
No. CBP300 are the proteins that are analogous to that accelerator pedal that sit on the androgen receptor. So they're what we call co-activators of the androgen receptor, and they're the ones that drive the androgen receptor, these two proteins. CCS 1477 is the inhibitor of that. So it's a chemical drug that we've made and manufactured that then binds to P300 and CBP and stops it from working, takes the foot off the accelerator pedal. So the two are different.
Thank you for clarifying that. There's a question about your trial, and obviously I understand, being still in it, you may be limited, of course, in what you're able to share at this point. But do you have a sense as to sort of, when you would be making public the sort of the results from the current stage you're in, what that sort of timeline looks like, and then really sort of the timeline of sort of, if things go according to plan. And of course, with all the caveats that Professor Hughes has told us about, the number of drugs that actually make it all the way through, what the sort of timeline might be before it's generally available in the UK to treat patients. And again, with the caveat that, of course, NICE would have to approve it, etc. But just, I think, sharing a bit more of a timeline for people to get a sense of what would come next for you.
Yeah. So the stage we're at the moment is that we're very close to establishing the recommended what we call the recommended phase 2 dose. So we've gone up that kind of staircase of doses that Andrew illustrated. We're at the point, we're at the top of the staircase, and we pretty much know the kind of dose that we think is going to be well tolerated and is beginning to show some very encouraging signs of clinical benefit that we can then take into the next phase of clinical testing, which is to really understand more about the efficacy effects of the drug, either given alone or actually in combination with some of the second generation anti-hormonal agents. So we're close to that phase already, and in terms of public disclosure of the data, that could happen, within the next year, when we've assembled the package of data to a point where we think it's ready for presenting to people. We then need to do some confirmatory studies in phase 2, and then we go into the much longer and larger phase 3 trials. We then have to seek regulatory approval at the end of those phase 3 trials, and as you say, also NICE approval and payer approval. So, I think that the timelines for actual having the drug on the market are looking more like sort of 4 or 5 years down the road from the point that we finish the phase 2 trials. So it's not a quick process. In some cases, drugs can be developed extremely quickly when very early on in their clinical development, they're extremely active. And quite often that can be in a situation like the one that Andrew illustrated, actually, the NTRK patient, where there's a molecular feature that means that drug is particularly active. And in those situations, you can skip various phases of the clinical trial process and get a provisional regulatory approval based on a much smaller cohort of patients, with a requirement from the regulatory authority to back that up with further data in a confirmatory phase 3 trial. So some drugs can be developed extremely quickly down that track. Others may take a little longer to get to the end game, if you like. I don't know, Andrew, whether you any further comments on that?
Nope. In the spirit of time, that was an excellent answer, Nigel.
Thank you, Nigel, for that. There's one question, I'm not going to ask it, but this is just a one I just wanted to sort of flag that we'll follow up. There's one gentleman who's saying that he has applied to be on your trial with the consent of his oncologist, and he hadn't heard back. So perhaps I'll just sort of wanted to let him know that I can follow up with him, and I can speak to you afterwards. And if where we might direct him for more information, we'll do that. So I just didn't want you to think we were ignoring you. Professor Hughes, I think this one's for you. There's a question saying a new drug to be tailor-made for every patient seems to be the ultimate objective. But is that sort of really possible, or are we talking sort of groupings of drugs according to molecular profiles, or what really do you think is the kind of end sort of objective that we would love to get to?
Yeah, great question. I mean, if you follow the precision medicine logic to its nth degree, you would arrive at one drug per individual, and the closest you get to that is a class of therapeutics called CAR-T treatments, where they are tailor-made to the specific molecular characteristics of the tumor. They've worked reasonably well in pediatric patients, children cancers, and hematological bloodborne cancers, but they cost a huge amount and are inordinately difficult to organize, but they do and can represent cure. I think that generally the perceived medical wisdom is that you don't actually need to kill every last cancer cell. So long as you can hit the majority of the cancer cells, what's called the majority clone, and you leave a small amount left over, then our immune systems, as you heard me say, are efficiently good at picking up foreign material and dealing with it. So not every last cancer cell does have to be killed, and not every different cancer cell that makes up a tumor has to be killed by drug A for cancer A, drug B for cancer cell type B, and so on and so forth. As long as you can kill the majority of cancer cells, that's the key. However, there's an alternative school of thought which says, drug treatment will actually become a thing of the past, because by the time it's got to metastatic disease, it'll almost certainly kill you. There'll be so many different clones within your cancer at so many different sites that mopping up everything with one or two or three drugs is unlikely. And so many of you will have heard on the news of the UK playing a pioneering role in using blood sampling to detect cancer before it spread. And by taking a blood sample, you can characterize not only that you have cancer genes, but also which organ it's from. So then we can turn the image onto the ovary or the pancreas or the lung and look with forensic detail at just that organ for where the very small tumor has started to grow. And when that's caught early, surgery and radiotherapy, if needed, can essentially cure you of cancer. It only becomes incurable when it's spread beyond the original site. So a lot of the Cancer Research UK monies are now, I think rightly, going into prevention and early diagnosis than they are actually late-line treatment.
Thank you very much for that. This is a question came around sort of the effects of COVID, and one gentleman mentioned he was actually after the first COVID lockdown advised to withdraw from a clinical trial. Did you find that affected your trial, and do you know, has that been the case with many other trials that you're aware of? Were they affected negatively by the COVID lockdowns?
Nigel, do you want to answer for our trial, and then I'll answer for all of the trials?
Yeah, certainly we did notice a significant reduction in recruitment to the trials. A number of sites were unable to take patients on. So patients that were on existing therapy with CCS1477, absolutely no issue at all. They were continued. But in terms of new recruitment, that certainly slowed down in the first lockdown. Certainly not seen that more recently in the second lockdown and subsequently. That in actual fact, I think things are really almost back to normal now.
And within Manchester, certainly during the initial COVID lockdown, all new patient recruitment was stopped, but existing patients carried on as normal. The only change that happened was that if a procedure could be what's called decentralized, in other words, done virtually, like checking someone's symptoms, any adverse events, that was done virtually. And it was only things that couldn't be done virtually, like a CT scan or like certain types of blood samples, that the patient was then asked to come to the hospital where clearly there was a heightened risk than doing things in their own home. By and large, after that first wave, things did then pretty much return to normal, because the clinical trial staff could be pulled back from the front line and work on the unit again. The only exception to that is trials that require some strange procedure. So I'm thinking of some trials that, for example, need an endoscope to be done. And because there's a huge backlog on endoscopy services now, then clinical trials that require strange procedures or, you know, invasive procedures still haven't quite returned to normal in those other types of trials.
Brilliant. Thank you very much for that, Andrew. And conscious of time, I think we'll probably make that the last one. One more just came in as I said that, but I don't think we have time, because I think that one might be a long one. I will do my best to answer that offline for you. So I just want to thank everyone for coming and for joining us today. I hope you found that insightful and helped clear up some of the kind of mist around clinical trials. To just really put everything into a larger context. It was through our collaboration with CellCentric that obviously, as Stephen touched upon, we spoke to a lot of patients as part of this work around clinical trials, and it was that which actually inspired us to do this webinar, because from the feedback that we got, a lot of folks saying they weren't sure about how they worked or sort of what the steps were. So this and some of the other work that we're doing is really sort of in response to that. So once again, just thank you for your participation in the work that we do and in working closely with us, because that helps us definitely figure out what we're going to do to support you, and also in terms of how we can try to work with industry and academics to bring things together and make sure that patient insight is really at the center of everything that's going on. So thank you all, and thank you to Andrew, Nigel, and Steve for your time today. It was a real pleasure having you. And if you all have time, we do have a quick survey after this session, including a question on if you have any ideas as to what you'd like to be included in next year's webinar sessions. Please do feel free to give us your suggestions, because they are really helpful. Thank you all, and have a great day.